I'd like to introduce Tiffany Lewis from the Center for Science in the Schools at Penn State. I'm also a co-presenter on this, and we're going to tell you about a little project that we did ever since kind of COVID started. Go ahead, Tiff.
All right. Well, good afternoon, everyone. Thank you so much for being here. We appreciate you attending. This session is going to be interactive as we discuss the creation of a set of online modules called The Science of COVID-19. We will have you engage in some of the activities of the course, so it will be helpful for you to either be able to access the modules on your computer or on your phone. And I think that's all I have to tell you to get started. So let's go ahead and get started. Like Matt said, my name is Tiffany Lewis, and I'm a STEM education outreach specialist at the Penn State Center for Science in the Schools, and we are actually located in Central PA. So a little bit about the Center for Science in the Schools. We are located in the College of Education at Penn State. However, most of our work is actually done with scientists and engineers at the university. The center is made up of former K-12 teachers with experience teaching science and/or engineering in the classroom. And our focus is to work with Penn State researchers on the broader impact section of their technical research grants. And of course, I use broader impacts loosely because it's a terminology for NSF grants, but we also work on a number of different federal funding agencies grants as well, like USDA, NIH, et cetera. And not only do we help the researchers author the broader impact sections of their grants, but we also implement their work as well. So the broader impacts work that we typically do is we work with the scientists and engineers, learn about their research, and then help translate their research to the classroom through teacher professional development workshops. So we'll bring teachers in, they'll learn a little bit about the content, and then we'll kind of help them co-develop some curriculum that they can use with students. So a little bit of background here on this course. Early on in the pandemic, obviously we know there was a quick switch to remote learning, and teachers were struggling to find free, high quality remote learning resources for their students. So this course was actually developed in response to a seed grant fund that was released from Penn State in response to the COVID-19 pandemic. Those funds were mostly meant for COVID-19 research. However, we saw an opportunity to develop a course for students, teachers, and the public to promote scientific literacy related to the pandemic, and also to provide a very relevant context for science learning. We were able to write the course in a way that engages students in the next generation science standards, science and engineering practices. So we're also developing a model for resources that can be created to improve STEM teaching through this course. This session isn't necessarily meant to be like a how to do this, but more so a look at what we did, how we did it, and then how something like this could be created for broader impacts work. And just a quick caveat, like I said, this was created from COVID seed grant funds from Penn State. So although this course is not necessarily developed as a broader impacts project, the science that was happening was in process and happening right now. So we wanted to turn it into something educational, which is of course something that is the tenet of broader impacts work. So creating online modules such as this in this way to tie current research into what students need to know for their courses in a way that's aligned with STEM education best practices. This is a great way to bring research to the classroom. So another interesting thing about our course is that it was developed alongside experts in the field. So we actually, as the science education faculty at CSATS, we wrote the content, but then we worked with experts at Penn State in virology, epidemiology, and public health, which are the three modules of the course, in order to check the course content for accuracy, of course, but we also wanted to know that it was authentic, like actually how scientists think about going in and exploring a novel virus. We didn't want to just throw something out there that was really focused on K-12 education. We wanted to think about it from the perspective of how scientists actually research this. So we actually were able to get guidance from them, and they gave us a lot of great feedback to help us fix our course to make it more in line of how scientists think about exploring a novel virus. As we developed the course, we were intentional about addressing some of the issues that we see in a lot of classroom instructional materials, even prior to the pandemic. We know that most content in science textbooks is knowledge that has been around for decades. And of course, it subsequently is treated as fact. In learning things like the scientific method, students view science as a series of steps, and that they think of it as a linear trajectory from point A to point B or C or whatever. This provides students with the misconception that science is a static and not dynamic field, which is, as we know, incorrect. And a lot of times we heard people saying that as the more information was coming out about the pandemic, that they were changing their minds and et cetera, but that's just how science works. As we find out more information, things are going to change. So this course is a resource for disciplinary learning through the relevant context and ongoing context of the pandemic. And like I mentioned before, we really strive to engage the students in the practices of scientists and engineers through a remote learning environment. And what I mean, sorry, I've already said practices a couple of times, but what I mean are the 8 science and engineering practices that are put forth by the Next Generation Science Standards, which is the current science education reform framework. So one of the interesting things about this course too is that actually some of the information has changed now that the pandemic has been in swing for a couple of years, and we actually developed the course in 2020 to 2021. So it gives an opportunity to have a discussion about how scientific knowledge is constructed and how it changes over time with new information and how new understandings come about and change. All right, so we're going to go ahead and pop into the course. So like I was saying before, we are actually going to have you go on to the course and register. So if you go to this website that's here, you can type it into your browser. You can actually just type in covid19.csats.psu.edu. Or you can actually scan that QR code with your phone if you'd like to access it that way. We made sure that the course was accessible through HTML and also through like a phone device or an iPad or whatnot, because we know that schools have different technology for their students, like either Chromebooks or iPads or whatever, depending on where they're at. So go ahead and go to that website now. And actually, I could probably drop this link into the chat too, right? COVID-19.
I will add, we are not selling your contact information. We won't bug you. This is just a way for you to log back in, because it takes roughly 3 hours from start to finish. So the only thing that your login is being used for is to remember that you've been there and to keep track of your certificates when you complete it. When it says that your progress is saved, it does, but it doesn't return you to exactly the same page that you were on when you left. If you pass a module assessment, then that's the information that's saved, but it doesn't actually return you exactly the same page.
And I also wanted to note too, you do want to register as a teacher, because teachers actually get access to some extra resources that I'll talk about in a moment here. Again, because we were former educators, so we know that teachers want resources to think about how to implement things, and also like handouts of things for their students. So we want to go ahead and make those for teachers. All right, once you get registered, it's going to take a minute because it does ask a couple questions, but if you could just give me a thumbs up or a little wave or some kind of reaction through Zoom, I'll know that we are ready to move on, but I'll wait here for a second. And if you have any questions about registering or whatnot, please let me know. All right, so once you get into the course, you'll see that, oops, I guess I could get out of here for a second. How do I exit? So you'll see that there's this dashboard here, and you have module 1, module 2, module 3, begin. So this is kind of your dashboard. This is where you go to get back to the course. And you'll notice up here too, when I was talking about, oh no, I must not have registered him being my husband as a teacher. Give me 1 second. Okay, there we go. So you'll see up here that there's these teacher resources, and if you click on the teacher resources, there is that implementation guide. There's a student and teacher version. The teacher version, of course, is the answers for the handouts for each module. And then there is a copy of the module quiz. So at the end of each module, there's a quiz about the content, and also that walks the students through different examples based on the content to be used for assessing learning or whatnot. So some teachers might use this for an informal assessment, but some teachers might want to use this for a formal assessment. So we did actually provide the quiz on here as a copy that they could print out and actually have their students do in class. So those are the teacher resources. It's not much, not crazy, but I think it would be helpful for teachers who are trying to implement this in their classrooms. All right, so we have 3 modules. The first one is based on virology, and that is focused on how researchers take an unknown DNA or RNA sequence and then characterize that novel genome to find out more about the virus. The second module is based on epidemiology, how modeling can allow scientists to predict spread or effectiveness of an intervention. In our case, for COVID, it was like masking, social distancing, things like that. Module 3 is based on public health, so how to anticipate hospital capacity issues. So are there enough beds for people? And then also thinking about system or county's rate of infection and things like that. Thinking about vulnerable populations. All right, so we're going to go ahead and pop in first to the first module, which is the virology module. So just a little bit more of an overview on this. So in lesson 1, the students work through identifying the virus first. So it's kind of set up as like a case study. So it says Mr. Lee, this patient, XYZ, whatever, came into a hospital with these symptoms. They're not really sure what the virus is that's causing it. So now we kind of have this unknown virus and we have to figure out what it is. So they take a sequence of the genome of the virus. And then once they figure out kind of what it's related to, then they can characterize the virus based on what's already known about certain viruses. So they'll come to find that SARS, et cetera. Then the second part of the module is about vaccines. So the students learn the basics of the procedures of vaccine development. So how is it possible to do that? And then also consider, based on their knowledge of the structure of SARS and things, SARS-CoV-2 and things about it, they could figure out or think about some things that could be targeted with a vaccine. So oddly enough, I did a focus on mRNA vaccines for this for this first portion of the course, which is cool because Moderna and Pfizer are both the first mRNA vaccines to actually be approved by the FDA. So it's kind of neat. Okay, so to get into module 1, you're going to click on that yellow begin button that you saw on the dashboard. And we're going to kind of skip ahead a little bit. So you're going to go to lesson 1, a new virus. And then you'll see there's a bunch of pages underneath of that. And you're going to go click on activity 1, identifying a virus. So once you get there, all right, so it's going to be this one right here, activity 1, identifying a virus. So it should look like this. You'll see a picture of this. You are a virologist. You have extracted the viral nucleic acid from the unknown virus. So at this point, we don't even know if it's RNA or DNA. And you use the sequencer to obtain the sequence of nucleotide bases of the unknown virus. Of course, obviously, there's some assumptions that students using this course kind of have a background of DNA, RNA, some biology concepts, but it does tie in this content to actually like drive it home. So students aren't just memorizing these things, and actually gives them a context to learn it in. So using the data, meaning the nucleotide sequences, how you go about studying the virus that has been isolated from Mr. Lee's respiratory tract cells. So you're going to click next, part 1, using sequencing data. And do one of my brave souls here want to actually share their screens and help us go through this? All right, so when you come here, you'll see that it says part 1, using sequencing data. The sequence of the unknown virus is actually here in this like orange box surrounding that nucleotide sequence. And this is, okay, this orange box here is the sequence of the unknown virus, which is actually pretty cool because this is a real sequence of the novel virus of SARS-CoV-2. I actually pulled it off of some sequencing data from BLAST. And then I actually pulled off some other sequencing data of some other viruses and put them on here. And these are the known viruses. So what students would do is they would compare the unknown virus and to these known viruses, and they would try to figure out, okay, what is this most similar to? Does it exactly match anything that we already know, or is it closely related and it's not necessarily a virus that's already known? Okay, so then there's these think like a scientist questions, which are kind of cool. And these questions actually like help students go through and think about it. They aren't just like questions like factual questions or recall questions, are more like thinking through the process of identifying a novel virus. So if you click on next, bioinformatics, this is the part where they're actually going to compare the unknown sequence to the known sequences. So there is a link here called Go to Fuzzy DNA Search. So if you go there, and there's actually a test sequence here, so we can go ahead and get rid of that because we don't want that sequence. And we want to go to copy the sequence of the first known. All right, so we want to first copy a sequence of a known virus. So in this case, we're going to do a sequence of the HA protein from influenza A and put that into our test sequence. And then the query sequence is the sequence that we're looking for, the one we're trying to identify, whether or not it's similar. So we'll take the unknown virus and we'll put that into this sequence here. So you'll notice that there's like some match values and things like that here. That's going to tell us how closely related these two sequences are. So you can take the top score here. So from nucleotide 1 to 25, the score is 7. So only 7 of those nucleotides actually match along that sequence. So if you come back to this page here and scroll down, you'll notice that you actually have a place to put in this data for the match score. So we saw that it was 7, and then we go through the rest of these, and we'd actually come to find, let's skip ahead to one of them. Let's try this sequence for our known. So I can get rid of that, add this in. And the score comes up to 18. So out of 25 nucleotides, 18 of those nucleotides match. So that's interesting. You come to find, if you did these other two, that this unknown virus is actually not known at this point, but it is most closely related to SARS-CoV, the S protein of that. So then we can do another check here to see if it's potentially closely related to SARS-CoV-2 by looking at it from the outside. So this is our this is our unknown virus here. This is the influenza A. So if you look at those two structures, these are very like oval, these are circular. If you look at this here, they're like, oh, those kind of match up, circular with some spikes on the outside. This one here, you can see there's some structures on the inside that are not present here on the left. And then the D has these like weird bubbly structures on the outside that again, don't really look like this one on the left. So if we had to say which one looks most closely related, we could say here that SARS-CoV, based on this electron micrograph data, makes it look most closely related. So at this point, we know that the unknown virus is closely related based on the nucleotide sequence, closely related based on what it looks like. So now let's do a final confirmation to see what virus we actually have. So if a scientist, like in the very early stages of the pandemic, were to take this unknown virus sequence and actually put it into this nucleotide blast site, they would have got no 100% matches. They would have gotten close matches, like 70 to 80% matches of SARS-CoV, but they would have gotten no 100% matches from this early on. But now, since we have a bit more data about this, let's see if we can figure out what this virus actually is. So you go to the blast site and you put your sequence into here, and then you click on blast. And it's going to give us, or take a second to give us the results here. Oh, there we go. Goodness gracious. Okay. So then if you're looking at this, like I said before, there would not have been any 100% matches on here early on in the pandemic, but since we have a lot of data about this now, like integrated into the blast system, we actually find that this is the severe acute respiratory syndrome coronavirus 2, which we know as SARS-CoV-2, which caused the COVID-19 pandemic. And we have a 100% match. So from this, students would be like, okay, well, we went through these processes to try to identify what this virus is. How do we get this 100%? Well, we now know that it's SARS-CoV-2. We've named it, all that jazz. But again, if we would have just been looking at this as scientists in a novel perspective, we would have gotten a match about 75%, because it's the SARS-CoV-2 is about 75% the same as SARS-CoV. So based on that data, we can make some conclusions about what we know already about SARS-CoV, and kind of make some conclusions about SARS-CoV-2, like how it's transmitted, what kind of cells it infects, so on and so forth. And then characterize the virus based on those things. So that was the first activity that would be good for students to go through to think about the virology aspect of the SARS-CoV-2 pandemic. So like I said, we uncovered that the novel virus is similar to SARS-CoV, so we could get some ideas about the virus from that, like it has a S protein, which is the spike protein on the outside, that it binds with ACE2 receptors to get into respiratory cells, so on and so forth. So we know some things now about the virus. And these are the kinds of discussions that teachers could have with students, like helping them think about, okay, in real time, science is changing. We found out some new things. Like at first about the pandemic, we didn't know if it was transmitted through droplets or just through the air, so on and so forth. So basically identifying and characterizing the virus gives us some of that information. Now, moving on to module 2, this is the epidemiology module. And in the epidemiology module, the students use an agent-based model to investigate the spread of the virus. And what an agent-based model is it simply means that the model follows the actions and interactions of the agents in the module, in this case, people, to see how they affect the system. And the system in this case is the population as a whole. The model that we developed for this course is a susceptible, infected, recovered model, also known as an SIR model. And a colleague of ours at CSATS actually developed the model in NetLogo, actually before he was a colleague of ours, which is how we scouted him, which is pretty cool. And NetLogo is a modeling software that you could create agent-based models in. And we developed this model, this SIR model, with assumptions already built into it that were based on current understanding of the spread of the virus at the time of the module's development. So again, that's pretty cool, bringing current research into the classroom. In module 2, the students explore the spread of the virus with no intervention. So how does it just spread through the population in general? Then they can compare the spread of the virus based on different interventions, including staying at home, social distancing, isolating when sick, wearing a mask, so on and so forth. Today in this session, we'll actually focus on modeling the spread with wearing a mask. So just a little quick more about the SIR model. This is an actual type of model that epidemiologists use to study infectious disease. So this isn't just something that we made up for the purposes of this activity and the purposes of this course. Epidemiologists use information on the virus's characteristics, mode of transmission, and genome sequencing to determine how the virus spreads throughout a population using this model. They also use the models to identify disease control recommendations. So based on what they see with how the virus can spread, that's how they come up with some of these ideas of things like social distancing, mask wearing, so on and so forth. Through using our model on this course, students can develop an evidence-based understanding of why certain measures were taken during the pandemic to control the spread of the virus. And then again, the SIR stands for susceptible, which are individuals who are at risk of becoming infected. So in the population, in our model, everyone starts out as susceptible. They have the risk of becoming infected. Infected individuals are individuals who have the infection, and if they come in contact with a susceptible person, they have the possibility of passing it on. And we have a certain probability of that being passed on built into the model based on the current research that was available to us when we created this model. And then the last part of this is recovered. So individuals who have had the infection for a certain amount of time then become recovered, and they can, in our model, can no longer be infected. But as we know now, that's not really true to the current situation, but models have limitations. Okay, so in order to get to the investigation for wearing the face mask, you would go into module 2, and then you would open up the N95 page, which actually has the model that we're going to be working with for module 2 for this investigation. And if you did a quick search on Google and looked at the percent effectiveness for different types of masks like N95, cloth, and surgical, you probably come up with different percentages based on the research that you look at. Tons of studies have been done since we actually created this module. So there's all kinds of information out there about the mask effectiveness percentage. Matt, did you want to add something?
No, I think that this is, again, another situation where you can have some really interesting discussions with students about what science is, how it works. Even if you develop a model and you program all the math correctly, it still relies on some assumptions and some other data that will continually get better as time passes. And one of the things that Tiff and I were talking about in developing this workshop is that, how do you test how effective a mask is? It seems fairly simple, but it's not, because you can test what percentage of particles make it through. Well, that doesn't necessarily work because it's tough to tell what is contagious and what's not. You could set up a randomized controlled test and give people that have COVID some masks, and people that have it would be in the control group, wouldn't have masks, and then you would count to see how many people got infected. That's not ethical. So the CDC website, when I checked this morning, has 90 citations of different studies that attempt at answering the question of how effective are masks at preventing COVID. And it's fascinating to see all the different ways that they do it, all of the different cohort studies, all the different ways that they try to answer that question. And at the end of the day, it's just not easy. So what you can do with this model is run it multiple times and kind of show what if the masks are 27% versus 95% effective. And it really changes the results based on what we know about the transmission. So Tiff, why don't you run it, and and if it's just you, me, and Jeff and Morgan chatting about it, that's cool. Let's just do it. Let's have a conversation about it. Yeah.
All right. So when if we were doing this as an activity, we'd have you search up the mask effectiveness. So I did a quick search this afternoon about the percent mask effectiveness, and I found that from an EPA site where they compiled some different studies, they said that cloth masks were 27% effective. Surgical masks were 39% effective, and then the N95 mask was 95% effective. And the EPA site actually had really interesting results compiled. They did all kinds of different things with studying the surgical mask, like flipping the elastic parts around, like some people do, to cross them, and then tucking in the sides, and that changes the effectiveness. So there's all kinds of different things that are obviously going to change the effectiveness of a mask depending on how it's worn, et cetera. But using this model, you can put in these different percent effectiveness amounts, and you can see how they would change the spread in the population. So going to the course and going to module 2, epidemiology. And it is underneath activity 2, N95 mask. this takes us to our net logo model so that we can run different percent effectiveness. And I realize I'm zoomed out kind of a lot, so let's zoom in a little bit. Okay, so you'll see that there's these sliders here on the side, which allow you to test different interventions like infection probability. I'm sorry, that's not an intervention. I meant mask effectiveness, move probability. So that has to do with like staying at home, social distancing. And then later on, there's also a button too that says like isolating when sick. So people who become infected, if they isolate and they don't go anywhere or see anyone, in effect means that they cannot pass the virus on. So you'll see that that causes some different changes in the model. So there's also a model speed up here. That's kind of nice too, because you can make it go really fast if you want to, if you're trying to click through this. So we're going to start out with the cloth mask, and that'll actually run the control at the same time, because you'll see here that they'll show you the control versus the treatment. Which in this case, we're looking at mask effectiveness. So the model's actually set at 95% right now, because I want to do cloth. What did I find? 27%. So I'm going to change my mask effectiveness down to 25%. And then to get the model set up, you actually have to hit set up. And now you'll see the little people here on the model. And you can see that one little pink person, that's the infected person that we're starting out with here. And then the rest of the people here are susceptible to the virus. All right, so then to start the model, you're gonna hit go. And you'll see here, we're gonna increase the number of people that are getting sick. Same thing over here, the gray people are getting sick on the control side. And if you let it run long enough, you'll find that there's a time when everyone in the model ends up being sick. So I can click go to stop that. And then you can see the number or the amount of time, quote unquote, number of hours it took for all of the people in the population to become sick. So if I scroll this along here, my XY coordinate shows me that 250. All right, so after 183 hours, quote unquote, 250 people were sick. So that's the whole population minus the infected person that we started with. So I would take that number 183 and I would put it in my data table up here. I did 27, 183 for my first trial. And then if I look at my control, that was with no intervention. It took 73 hours for 250 people to become sick. So just in that first comparison there, you can see that with a cloth mask, even though it's only like 27% effective, it controlled the spread of the virus through the population a little bit longer. So if you were continue to go through these trials, this would calculate an average for you. And then you would see that, you would see that in the end, that the N95 mask, being that it's 95% effective, is going to take the longest amount of time for everyone in the population to become sick. So it's kind of interesting.
Can you run them?
Run them all?
Yeah.
Okay.
I don't think you need to do all 3 trials for all, but
at least run each one.
Yeah, let's see what we get.
All right.
39%.
So we're gonna go to, and the nice thing is you don't actually have to slide it. You can just type it, which is easier. And then you hit set up, make sure it says 39, we're good to go. And then we're gonna hit go. I should have sped the model up. You can't retroactively speed the model up, which is sad.
We're getting there.
All right, looks like we hit it. Sweet. All right, so 237 hours, 250 people became sick.
Now, these aren't real hours. They're ticks on the modeling program. So it's not, we're not exactly claiming hours, but That's the unit, if we're going to compare units of time.
Yeah, for the model itself. Okay. And then the last one is going to be the N95 mask, which we identify was 95% effective. So I come back over to my model.
Speed it up.
Yeah, definitely speed it up for this one. All right, set up and go. So as you can see, this line is staying way low for a while since the N95 mask is 95% effective. So we even have our control people becoming recovered before we actually even hit all the folks in the population. And if you let this go for long enough, you can see there's some kind of interesting stuff going on here. Because notice in the treatment, now you have 3 different types of agents in here. So we've got the pink, which are the infected, the yellow, which are susceptible, and then the green, which are recovered at this point. So if I just stop this, because almost everyone has been infected and recovered at this point, looking at this model here, you can kind of see that there's never a point where the entire population became infected. So like, what does that mean? Like, why do we never hit that plateau? That's an interesting conversation to have as well with students.
Let's just put it out to the group. Any thoughts on kind of discussions that you could have with students about these models?
Jeff, you want to throw something out there for us?
Yeah, I mean, these models are very interesting. I did a thing that was an NIH lab for middle school at a much lower level called Protecting the Herd, which kind of did the same thing on a very simple middle school level, where you just did a whole classroom and you changed the variability of who would be infected. And it was sort of a paper simulation that was done. And then after it ran its course, you had the kids raise their hand who had immunity, who were susceptible, the same kind of parameters. This just gives you a lot more detail and variability and controls. I mean, you can go a lot of different directions with this compared to that simple middle school activity, but it's essentially the same kind of element.
Yeah, and I think one of the early discussions we had during the development of it was this idea of adding a control side, because one of the frustrating parts for a lot of people is, there was a lot of complaint early on that there was, the state or isolate, for the 2 weeks to flatten the curve. That was kind of the battle cry, right?
Right.
And it's tough to tell whether it worked or not, because you don't really know what would have happened without.
Yep.
And so this at least gives users a way, okay, if masks are really 25% effective, is it worth it? Is 25 a lot? Because you're running a simulation with several people, over a long period of time. You see that, I would say that there is a fairly significant, effect in the number of people that get sick and the amount of time that it takes everybody to get sick in that population. There's lots of ways to run this kind of model, but it definitely opens up a lot of potential discussions you could have in ways that, aren't typically done in a life science class, I would say.
Yeah, and for sure that the control is a good element. I know kids always coming to me from elementary, the focus was you're only going to do one variable at a time. Realistically, the world is full of lots of different variables. You can't realistically control for one variable all the time.
Yeah, good.
Yeah, for sure. All right. And then, so that was module 2. And another important conversation I think to have with this is also the limitations of models. So looking at all of the things that are can't be accounted for or are not accounted for in this particular model is important as well. And again, like I said before, we developed this model based on the data that we had at that time for infection probability and so on and so forth. But as we know, science changes over time, and there's all kinds of new information that has come out about the virus since these were developed. So students may find some things in the model that don't seem quite right. And that's definitely a possibility because, again, there's limitations in models, and then sometimes things in models don't always pan out the way that it predicts it should. So a lot of interesting points of conversation for students here. So the final module is module 3. This module is focused on public health, and it goes through kind of the thinking of public health scientists. They need to understand how to analyze data, develop public policies based on that data. They have to be able to communicate with many people from a wide range of fields, from scientists to the general public. They have to be able to manage people and help them kind of do these interventions. And they also have to be able to think about interpreting complex systems. Obviously, populations, humans, huge complexity there because we can't control people and tell them what to do. So we have to think about all the possibilities and kind of put that all together and thinking about the system. And in this case, the spread of the virus. Another thing that they have to focus on are vulnerable populations. So why are they vulnerable and what can we do about it? That was definitely an interesting thing that we saw in the pandemic. Early on, we saw that there was a lot of like inequities in healthcare, as there always are, but that consequently had COVID's impact had more of an impact on different populations based on their vulnerability. So that's kind of what this module focuses on. And one cool thing about this module in our course is that with the help of our web developers, we are actually able to populate actual data about the number of infections in particular areas into our model that shows public health. So this is super important for students because a lot of the time we only get data from like big urban areas, and like looking at this map here, you can see Pittsburgh and Philly are our two big urban areas in Pennsylvania. Harrisburg a little bit. It's the capital, so that's why it's on there. But all the students that are in between here, there's a huge gap where there's probably not a lot of data available about the infection, the pandemic. This in the pandemic, in our case, is a little different because there was a lot of people reporting on that data. But typically we only get information from like urban areas. So the students actually were able to search up their own area, their counties, get real-time data, and allows them to explore the pandemic in their particular context. So part 1 of the public health module looked at healthcare capacity. So this is kind of related to, this is related to flattening the curve, which most likely you've heard about at the beginning of the pandemic. So what the flattening the curve actually means is that we were taking measures to control the spread of the virus at such a rapid rate with no intervention at all, so that healthcare systems were not overwhelmed with patients and it didn't run out of the capacity to help. Some of the things that this module explores is the number of hospitals in a certain area, so you can search that up. The number of staff beds, the bed utilization rate, meaning how many of the beds are typically occupied, and then also the amount of capacity that the hospital has based on current numbers. So again, we're pulling in data, real data that actually helps you look at those different things and explore how the pandemic impacted your particular area. So like looking at flattening the curve here, we wanted to make sure that the healthcare system was not overwhelmed. So we did try to use all those protective measures, especially early on with like the 2 weeks of, well, it wasn't just 2 weeks, but the staying at home orders and things like that to try to decrease the number of people getting sick at the same time so that all of these severe illnesses were not all in the hospital at the same time. So one of the challenges with creating instructional materials and courses like this based on current knowledge and unfolding knowledge is that the more knowledge that's constructed about a particular phenomenon, in our case, COVID, the more the information changes. So with our public health module, we found that there are some things that aren't quite the case anymore. So we are trying to pursue more funding so that we can update those modules to reflect more current data. But I can kind of explain how we developed the public health model by incorporating real-time data. So thinking about this bed utilization rate, it's a decimal to represent the fractions of beds that are typically in use at a hospital. So you can pull up this data, and I actually got this data from, I think, American Health Association. Is that what that stands for, Matt? Or Hospital Association. Sorry, just kidding.
Yeah.
And we found that according to them, that there's actually 2,814 hospital beds, but depending on the sources that you look at, that number might change as well. But at the time that I pulled this data and the week ending, well, I didn't pull it the week ending here, but the most current data they had was the week ending of January 13th, 2022. There were 1974 hospital beds occupied, and 624 of those beds were occupied by COVID patients. So you can figure out the bed utilization rate by subtracting those percentages. 70% minus 22% gives us a bed utilization rate of 0.48.
And if I could just add a little bit more context.
Yeah.
When we first designed this, there was a simple link that you could search by county, and then it had a little dot for every hospital that was in that county. And one of the quick statistics that popped up in a little box was bed utilization rate. So that's why it's in there. When we get funding for extending this course, we will modify this section for sure. But that's kind of where bed utilization rate came from. It's really about a way to kind of predict how many available beds that we have that can deal with the pandemic, on top of the stuff that you already are going to have with people having heart attacks or pneumonia or these other illnesses or other reasons why people are in hospital beds to begin with.
All right. And then, yeah, another thing I was going to say too is, like, again, using current situations for instructional resources, again, like in this more like semi-permanent type of course, is that, again, the resources that you draw from are going to change too. So, there was a lot of these like statistics websites and stuff like that that were coming up and popping out for us to see like real-time data about the pandemic. But now, since the pandemic is kind of waning a little bit and there's not as much attention on it, obviously those resources aren't going to continue to exist. So that's a challenge too, thinking about pulling current research into the K-12 setting or in an informal setting, even so, is that these resources might change. So we try to not go out to the outside sources as much as possible. But of course, like when you're trying to pull in real data, you have to go outside your course to do that. So the part two dealt with a prevalence curve, which is the total number of people that are sick at the same time that would need to be hospitalized. So this is that curve you were kind of seeing with the flatten the curve type thing. How many people are sick at the same time? And what is your healthcare capacity in your area? And were those hospitals becoming overwhelmed? So we, with our help of our web developers again, actually created a model that you can put in data about your county, and it actually develops a prevalence curve for you. And that information that's in that model had to be calculated, obviously, but it was also based on some assumptions as well. So at the time that we created this course, the data said that severe cases make up about 5% of total cases. So that was pulled into our model. And then also severe cases take an average of 10 days to resolve. So again, that was also pulled into our model. And
I'll say that our website is designed to scrape the data from USA Facts every morning. So every morning, it brings in information about new cases, and then just through some fairly simple arithmetic, is able to generate these curves. So they will continue as long as we allow it to. Obviously, a year ago or a year and a half ago, these curves looked a lot different because we haven't gone through different waves. But it's actually interesting to look at different counties.
Mm-hmm. And then we have a sandbox that was incorporated into the course where you can search up your own county and create a prevalence curve based on your county. So I actually did this the other day. And here is I didn't include the data necessary to get a hospital capacity, but I have my prevalence curve of these different peaks of the pandemic. And like Matt was just saying, there's these different peaks that are in the data. And that's an interesting conversation, because if you look at this data, the highest peak of severe cases, quote unquote, was happening like early this year. But you got to think about the context of this as well. So what was going on at the beginning of this year? What would have caused this big spike in COVID cases?
Christmas gatherings.
Christmas gatherings, and also
Omicron.
yes, very good. So Omicron was a more contagious strain of the virus, but wasn't necessarily as severe in causing sickness. And also too, we have people that are vaccinated now that are getting less ill than they normally would. So it's not truly that these cases were all severe enough to be hospitalized. So again, that's another limitation of this particular model, because we don't have those types of things built into the model. So it's showing that these people had severe cases, but it's probably not the case. We know that the hospitalizations were not that much as they were early on in the pandemic before vaccinations and things like that were happening.
Hey, Tiff, can you go to the sandbox? Let's just do a couple
Yeah, sure. Okay, so to get to the sandbox, you go to module 3, and then it's underneath activity 2, developing a prevalence curve, and here it is, sandbox. So it's called sandbox because you can go in and mess around with it without having to actually go through all the steps of the module.
So Jeff, pick any county in the country.
Lebanon County.
Lebanon
Boo.
County.
I was going to say bonus points for outside of Pennsylvania.
Do you know how
Okay.
many? Do you know how many hospitals there are in Lebanon County?
There would be 2.
2. All right.
Yeah, and I think we certainly had our time of maxing them out.
Okay. And then add row, we don't know the hospital names, but
Well, I'm sure Jeff does.
Yeah, Good Samaritan and Penn State College of Medicine.
that's in Lebanon County.
Oh, sorry, 1. Yeah, no. Yeah, no, my bad. Yeah, Lebanon, it's Good Samaritan is the only one. Sorry about
Okay. No worries.
that. I mean, most people in Lebanon County go to Penn State. We don't go to Good Samaritan, so it's like that's our default.
Yeah. Matt, are you looking at staff beds?
I am looking it up. Yeah.
Oh, you are beautiful. Thank you.
Okay, so it says that Lebanon County has a total of 111 staffed beds.
Okay.
And then let's estimate a 50% utilization rate.
Alrighty. So I put 0.5 for that. All right. So now I'm going to click save data. All right. So
So I think that this probably is fairly accurate. Jeff, would you say that Lebanon or the Good Samaritan was having some capacity issues like at the end of 2020?
Oh yeah.
Yeah. And then
Oh yeah.
And then I think, again, like Tiffany had mentioned, the Omicron kind of threw off the assumptions from this model. And I think when we do the redesign on this, one of the things that we'll be able to do is to have a slider or an entry to change that percentage, that assumption, because, I think a significantly smaller population of people were hospitalized with Omicron because it doesn't involve the lungs as frequently as Delta and the previous ones. But again, these are discussions that can be had. I think some of the other discussions that you can have are like what's happening now. And so it's a matter of looking at the graph and kind of tracking trends. What do you see now in Lebanon County, Jeff? Based on this graph.
Yeah, based on that graph, I'm having a hard time seeing where the dates are. The most
Oh, this
current
is
is
like
Yeah, the
it's
last date all the way
oh
on the
yeah,
right is today.
right. No, we're low, but it's slowly ticking up, and
Yep.
I feel that from experience sometimes.
Right. Right. And then, some of the other things that you could bring up, again, that helps data analysis, helps graph reading, would be like, what are the three highest peaks, of COVID? And you could, have them estimate the times, or you could say when were the lowest times, after May of 2020, that kind of stuff. You could have these kinds of conversations. Are we trending up now? When were our hospitals most in danger? Like, those are the kinds of discussions you can have that really beg for some analysis and some reasoning, which I would say that most teachers that I've run into would like more opportunities to find that in the class.
Yeah, absolutely. It's good stuff.
all right.
Let's do one more. Let's do some faraway county that maybe you've been on vacation to.
How about
Harris, how about we have a request for Harris County? Where's that, Missouri? Morgan?
No, it's I think it's in Houston somewhere.
Okay, Harris County, Texas.
Yeah.
Cool.
Whoop, there we go. Okay. Do you have any idea how many hospitals?
I cannot
I can
tell
look it
you.
up.
Oh yeah, it doesn't matter actually, because you can just look up the staff beds. So I need to remove this first, add a row. You can just jump to the staff beds. That's nice.
Okay, Harris County, Texas. I got it. Maybe it's trying. 9,875 staffed beds.
Whoa.
And,
All right, should we
yeah,
just-
just estimate like 60% or whatever, just because it's something that changes.
Yeah. Maybe I'll estimate lower or higher. Will that give us more peaks?
That's yeah, I don't know. That's really surprising. I think that Texas also doesn't report as much. I
forget which way I have to go. Oh wait, I can't. What was the number again? 9,875.
Oh, yeah, 9,875.
All right, now we got some better peaks there.
Yeah.
So kind of similar trend.
I think it says Lebanon County at the top.
Oh, it does. Why? Oh, what the heck? Son of a gun.
You got to change the state first.
Yeah, I didn't hit save data, junk, user error. Let's try again. All right, save data. There we go. Now we're popping. All right, there we go. Oh, okay. Add row.
9875. Yeah.
All right, boom.
So it looks like there were significant issues, at basically 3 times, and it's tough to kind of interpret what was going on in Omicron. I think some hospitals were still getting stretched pretty thin just from sheer number, but it wasn't quite as bad as the peak looks like. Does that sound like anything you've heard before, Morgan, or do you just kind of have been to Harris County and just wanted to see?
Oh no, so I'm actually from there, but I have not been back in a while. So yeah. So that's just the first county that came to my head.
Oh, that's great.
But I'll be back next week to see.
So I think, from the standpoint of, I mean, most of the kids, if you were teaching a class in a high school, most of the kids likely would be, from a fairly limited area. Whether they grew up there and stayed or moved once, but I mean, it's not like you would have an entire wide range of places, but you could certainly, bring in, I have a relative from, or I go on vacation to, or I've read about, or, you could have them look it up even. And you could really kind of, if you overlaid these on top of each other, I think even though the peaks would line up in terms of when they happened, generally, I think that, from even from Philadelphia to central Pennsylvania, where we are, is a couple weeks different. When their cases are starting to rise versus when ours do, or when, ours does versus other places across the country. I think the timing is different, and it's actually kind of interesting to think about that kind of stuff and discuss that.
All right, very good. Any more, Matt? Or
No, I mean, unless anybody has any requests.
San Diego, I'm gonna fly over there. Okay, so to kind of wrap this up, thinking about how this course addresses broader impacts work. Obviously, broader impacts is aiming to do several different things, but thinking about bringing current research to the general public, a course like this based on current research is a super great way to make that happen. In our context, we actually have been working with other researchers on their technical research grants. And some of the things that we are proposing are more courses like this, more online modules that would be able to translate their research to the general public, whether it's K-12 education or just users in general. And especially for those who do things like computational work and stuff like that, like not super heavy that requires supercomputers, but things that require some computing and things, those are very well disseminated through these online modules. So we're thinking about how we can expand out this type of situation and this type of course in the future. And are we even like looking at different course platforms and things like that, because we use web developers from Penn State, which is super expensive. That would be hard to pull into a technical research grant budget. So some of these other online platforms, I think we're looking at one that's called Articulate 360 right now. You do have to have a subscription and things like that, but you can actually create the online modules yourself using that program. So it's much less expensive than to pay like web developers and things like that. So it's pretty cool. Matt, anything else you want to add?
Just that so far we've had 3,241 people register, which is a lot more than we expected, but also kind of speaks to how normally we do teacher workshops that, have 20 teachers, and maybe if they do it with their students, you get a couple hundred students involved, and now knowing that we have over 3,000 in a little over a year is kind of exciting from a dissemination standpoint.
Yeah, we had I was looking back through my emails today to find something for this, and I saw that we had like 1,000 in 3 weeks or something, which is pretty crazy. So that's cool. Just real quick, like a little bit more detail about how we designed this, and thinking about, we didn't just go from like creating the content and then throwing it out there to the world. We had to submit the proposal, which we did in April of 2020. Then we found out that it was funded after some negotiations in May. And then in the summer, it took a couple months actually for us to develop the course content, then get in contact with the researchers to get their feedback. And we kind of had to go back and forth a couple of times to make sure everything was accurate and authentic to the discipline. Then in fall of 2020, we turned over the course to the web developers, and we actually developed it. I mean, you can do it in different ways, but the web developers that we worked with said you can just put the pages in PowerPoint slides. So basically each page was a PowerPoint slide that we created, and then we sent over to them, and they turned it into the course that you saw today. So it's pretty cool how that happened. Then in October 2020, we actually, we threw out a call for teacher pilots through our listserv. We sent out some stuff through the National EPIDEMIOLOgy Teachers Association on Facebook, and a couple other different places. We had a call out for teachers to pilot the course. So we chose 10 teachers to pilot the course and provide feedback. In the end, how many teachers do we actually have piloted, Matt? Was it 7, something like that?
8, I think.
Okay, yeah, 8 teachers that actually made it through. We had meetings with them. They provided feedback written, and they also had a meeting with them to follow up on that, to ask them some questions about the course. So we did take in feedback from teachers who are currently teachers, even though we were former educators ourselves. It's been a few years since we've been in the classroom. So we got that feedback and then made some revisions based on the feedback. And then actually the final course revisions with the web developers took about 3 months, November to January. And then early February, the course is actually ready for dissemination. So once we got the green light, we sent it out all over the place on our listserv, different places, shared it out on the some Facebook groups, national organizations, etc. And that's how we compiled all of our users right from the start. So it was a process, but I think it was definitely a super fruitful process. And in less than a year, we had a pretty solid course about the science of COVID-19. So we're pretty excited about this work, but we are more excited about potentially getting some funding for future plans to build out the course. So with some additional funding, we hope to update the course, obviously, to reflect the current information that's come out in the past couple of years. But we also want to expand the modules to include more curricular connections. So the first module, the virology portion, there's some really cool stuff with modeling of the different strains of the virus. So you can see there's Delta in here and Omicron. So we want to focus on the role of natural selection in the development of new viral strains. Also for the epidemiology module, module 2, we want to be able to integrate a map into the model to actually simulate population density. So here's a map here that shows the more dense areas of population. Right now, we just had a random assembly of agents on the model, but it'd be really nice if we were able to actually look at population density and how that affects viral spread, because obviously it was spreading in cities faster than it would spread in rural areas where people are less likely to come in contact. And then in module 3, the public health, this one's really cool. Students would be able to model the spread within a school by overlaying an agent-based model on a blueprint. So over here, it's kind of hard to see, but you can see this is actually a school building here. And you can see that there's areas where students would be. And by looking at areas where a student would be and thinking about how, and modeling that spread through the spaces of the blueprint, students can actually consider thinking about school policy decisions that would balance the trade-offs between what works and what people would likely do. So where are people at? Where will they likely be? Could they change the flow of how people walk around the school? Could they change the way that students get their lunches? Could they change the way that the cafeteria is set up? So on and so forth. So modeling all these different things to think about how they could decrease the spread of the virus in the population. So we have some really exciting plans for the future of this course. So hopefully we can get some funding to make that happen. So yeah.
That's the plan. We just need to find some money.
Yeah. So do you have money for us? That's our question. Just kidding. So there's our contact information if you have any questions in the future and want to reach out to us. And we wanted to thank our folks that helped develop the course, the researchers and the web developers, and also the pilot teachers, all who had a very important role in the development of this course. So again, thank you so much for attending today.